Greetings to everyone. Welcome back to this channel on biology. So I'm going to teach on dihybrid inheritance in this video, and we'll be discussing subtopic 11.2 from the Form 5 textbook. This topic is about inheritance. Now, following the good response to the video on monohybrid inheritance, I've decided to do this video on dihybrid inheritance, prepare this video so as to help you to delve deeper into this topic and understand it better, right? So just follow along, view this video, and I'm sure that you'll be able to answer questions on dihybrid inheritance with the understanding that you have. So Mr. Tall Purple married Miss Dwarf White. What will their children look like, right? So this is related to dihybrid inheritance. The learning standards for this video are as follows. Firstly, we should be able to describe the meaning of a dihybrid cross. Secondly, explain a dihybrid cross based on Mendel's experiments. Thirdly, construct a dihybrid cross diagram. Fourthly, determine genotypic and phenotypic ratios of phylogenerations in a dihybrid cross. And fifth, describe Mendel's second law. Now, our first learning outcome is to describe the meaning of a dihybrid cross. Okay, so here we define dihybrid inheritance according to the textbook. So it's related to the dihybrid cross. Okay, so what's the meaning of the word di here? It means two. Two what? Two characteristics. Okay, so dihybrid inheritance involves the inheritance of two different characteristics which are studied together at once, or we say studied simultaneously. Okay, so each characteristic is controlled by a gene located at a different locus from the other gene that controls another characteristic. Okay, that means for characteristic number 1, it is controlled by gene number 1, which is located at a locus or location on a pair of homologous chromosomes, right? And then there's another gene for that controls the other characteristic, but it is located at a different location from the first gene. Okay, and it is on another pair of homologous chromosomes, different type of chromosome, okay, from the first one. Now, as two different characteristics are involved, it means that two different genes are involved, because each characteristic is controlled by a gene. So now you have two characteristics, you have two different genes. For example, gene 1 for height and gene 2 for flower color. So maybe these two characteristics are studied together. The inheritance of these two characteristics are studied together at the same time. Now then, each gene consists of a pair of alleles. So now we break up the gene into its alleles, the different versions or different forms of a gene. So actually an allele is just a specific gene that controls a specific trait. Okay. Now, for example, gene 1 for the characteristic of height consists of two different alleles. We can assign the letter big T to represent the dominant allele for tall stem trait and small t to represent the recessive allele for dwarf stem trait. Okay. Take note that we don't write big T represents tall stem trait. No, it's the allele. It represents the allele, okay, which is a specific gene, okay, for tall stem trait. And small t is the allele for dwarf stem trait. Secondly, there's gene 2 for the characteristic of, say, flower color. So it consists of R, big R, dominant allele for purple flower, and small r represents the recessive allele for white flower. Okay. Thus, in dihybrid inheritance, two genes are involved or two pairs of alleles. Okay. So 2 times 2 equals to 4. So there must be a total of 4 alleles involved. So when we write the genotypes of an individual in dihybrid inheritance, the genotype must consist of 2 pairs of alleles, okay, or 4 alleles. For example, big T, small t is one pair of alleles, and big R, small r is another pair of alleles found on another type of chromosome from the first one, where big T, small t is located. Okay. Then in this genotype here, okay, going back to the first genotype, this genotype shows that the organism is heterozygous for two characteristics. okay, having one dominant and one recessive allele for two characteristics. But for this genotype, big R, big G, small g, so it could be homozygous for one characteristic, homozygous dominant for one characteristic, and heterozygous for the other characteristic. Okay, so alleles can be dominant or recessive alleles. Compare with monohybrid inheritance, only one gene or one pair of alleles is involved. So, for example, big T, small t, big G, small g, big R, small d, small d. So only one pair of alleles or two alleles are involved. Okay. Now this diagram illustrates the dihybrid cross, which we have, or dihybrid inheritance, which we have just discussed in the previous slide. Okay. So di meaning two. So in dihybrid cross or dihybrid inheritance, two characteristics are studied together. All right. For example, characteristic 1 may be height, and characteristic 2 may be flower color. Okay. So let's say we take characteristic 1, height, controlled by gene 1, which is gene for height. So this characteristic of height consists of two variants, all right, which is called the trait, which are called the traits. So first you have the tall trait, and secondly you have the dwarf trait. All right. So the plant can either be tall or dwarf. Now for the tall trait, it is controlled by the allele big T, which is a dominant allele, right? And for the dwarf trait, we can assign the letter small t to represent the recessive allele. Okay. So these two alleles are involved in the gene for height. Now, what are the possible combination of alleles in a diploid cell? Because a plant will have two of these alleles in each diploid cell. Okay, one from each parent, right? So what are the two possible alleles in a diploid cell? It could be two dominant alleles, big T, or one dominant, one recessive, big T, small t, and another one is small t. Because in a diploid cell of the plant, you have two alleles for height. Okay. Now, for the characteristic of flower color, it's the same thing. By the way, if we are just talking about one characteristic, then it's called monohybrid inheritance. Now you are discussing, or you are studying two characteristics together at once, the inheritance of two characteristics. Okay, so it's dihybrid inheritance. Now for characteristic number 2, which is flower color, it's controlled by the gene for flower color. And the characteristic consists of two traits, two variants, two traits, which are purple flower and white flower. So a plant can either have purple flowers or white flowers, right? So the purple flower trait is controlled by the allele for purple flower, which is R, big R. And the white flower trait is controlled or determined by the allele for white flower, which we assign the letter small r. Okay, and it's a recessive trait. Okay, right. So what are the possible combinations of alleles in the diploid cell for the characteristic of flower color? Okay, so again, each plant will have two alleles in the diploid cell for flower color, one from each parent. So it could be big R, small r, or small r, small r. These are the only combinations that you can get, okay, for the pairs of alleles in a diploid cell. Okay, so in dihybrid inheritance, when we write the genotype, we have to consider the height of the plant and the flower color. So you compare, you have to sort of combine the two genotypes, genotype for height and the genotype for flower color together. So for a plant that is homozygous dominant for height and homozygous dominant for flower color, that means homozygous for tall stem and homozygous for purple flower, then you will have the genotype big T, big R. Okay, so each one of these genotypes can combine with any one of these genotypes here, depending on what the genotype of the plant is, or what its traits are. That means you have 3 combinations of alleles here for height, combined with 3 combinations of alleles for flower color. So you get 3 times 3. There are 9 possible combinations for dihybrid genotypes. Okay, so this is just for our knowledge. There are 9 possible combinations for the hybrid genotypes. Right, so now let's look at how the alleles will look like in the cells. All right, so the alleles are carried by the chromosomes. Okay, they are genes that are carried by the chromosomes, and this is a pair of homologous chromosomes. Let the blue one be the paternal chromosome and the pink one be the maternal chromosome, okay, of the same type. So you have one allele on the paternal chromosome and one allele on the maternal chromosome. So this would be the appearance of the cell for the individual or organism that is heterozygous, having one dominant allele and one recessive allele in its cells, in each of its diploid cells, right? Then during meiosis, when the homologous chromosomes separate from each other in anaphase 1 of meiosis, then the alleles will separate too. Okay, so big T allele will be separated from small t allele, and each one will be found in a gamete. Right? Now, by the way, normally in meiosis, anaphase 1, each chromosome has duplicated to become two sister chromatids. So it won't be single-stranded like this, but be X-shaped, like, right, consisting of two sister chromatids. Okay, but the two sister chromatids are genetically identical, so both of them will have the small t alleles here, and this one, the sister chromatids, will have big T alleles. So whatever it is, finally, each gamete will contain only one allele, okay, from the pair of alleles in the diploid parent cell, right? So these two alleles will separate from each other. Now, how about in the hybrid cross? How would it look like in the cell? So now you have two pairs of chromosomes which carry the two pairs of alleles. So like big T, small t is carried on the first pair of chromosomes, homologous chromosomes. So let's say this is chromosome number 1. Okay, the type of chromosome is chromosome number 1. So in this pair of chromosomes number 1, you have these two alleles, big T, small t. For an organism which is heterozygous for this characteristic, maybe characteristic of height. But how about for the other pair of alleles? All right. So the first assumption we have to make is that the other pair of alleles is not on the same pair of chromosomes as the first pair of alleles. It must be on a separate chromosome, maybe chromosome number 2 or chromosome number 3 and so forth. All right. So the assumption for this, the hybrid cross, and for Mendel's experiment is that both pairs of alleles are on different types of chromosomes. Okay, they are not carried on the same chromosome, like big T here and then big R here, or small t here then small r here, something like that. Cannot. They are on different pairs of chromosomes. Then Mendel's second law will work for it, can be applied to it, okay, to apply to the formation of gametes. Right? So okay, now let's say you have the pair of alleles big T and small t on chromosomes number 1, and a pair of alleles big R, small r on chromosomes number 2. So this individual will have the genotype big T, small t, big R, small r. That means the individual is heterozygous for the two characteristics, for height and also for maybe flower color. Okay? Now, then what happens in meiosis? Simple, you see? Now, you can see this cell. In this cell, we didn't show the chromosomes. We just show the alleles. All right? But imagine that each allele is on its chromosome. So in metaphase 1 of meiosis, there's one possibility that the chromosome carrying big T, okay, this one, the pink chromosome, faces the same pole as the chromosome carrying big R. Okay? Then, whereas on the other side, small t and small r, the chromosome carrying small t and the blue one, and the chromosome carrying small r, the short blue one, the long blue one and the short blue one are facing the same pole. Okay, that means the paternal chromosomes face the same pole. The blue chromosomes face the same pole, and the pink chromosomes face the same pole. The maternal chromosomes face the same pole. So they will separate in such a way that they are together. Big T and big R will be together, small t and small r will be together. Okay, so the gamete form will be, the first combination will be big T, big R, both from the maternal chromosomes. And for the other side, both on the paternal chromosomes, alleles on the paternal chromosomes will separate and move together. What I mean, they don't separate, but they will separate from the others, the counterparts, and move to the same pole. So they'll be together in the same cell. So this is the second combination of alleles in the second type of gamete. So this one, first one, and number 4 is accounted for. Now, how about other possible gametes? Yes, there are other possible gametes. So instead of, now look at these two, the second arrangement of the homologous chromosomes at the metaphase plate, right, is that the big T allele on the pink chromosome, okay, it faces the same pole as the small r allele on the blue chromosome. So this chromosome and this chromosome faces the same pole, and on the other side, these two will face the same pole. The chromosome carrying small t and the chromosome carrying small r, big R. Okay, so this is different from this one, because this one, big T, small t, then big R, small r. But here you have changed place. Here the small r is together with big T already, and the big R is with small t. Okay, and that's where you get the third combination, big T, small r. Okay, on this side, these two alleles will come together to be in the same cell, and the fourth one is small t, big R. Okay, now it seems a bit confusing, but if we apply Mendel's second law, then we can easily determine the gametes without having to imagine how the arrangement of the homologous chromosomes are like at the metaphase plate during metaphase 1 of meiosis. Okay, now the easier method is follow Mendel's second law, okay, which is during meiosis, in the formation of gametes, each allele from the first pair of alleles, each allele from here, can combine with each allele from the second pair. That means big T, you see, this is my linking method. Big T can combine with big R, so you get this one. In the formation of gametes, you get big T allele coming together to be in the same cell as big R. Okay, or big T can combine with small r, you get the second pair. Now another combination is the small t allele can combine with big R, and the small t with small r. See, you also get the same results. All right, so that's Mendel's law, which we'll discuss later. That is, during meiosis, in the formation of gametes, any member or allele from the first pair of alleles can combine with any member or any allele from the second pair of alleles. So the linking method is 1 with 3. 1, 2, 3, 4. Let's say this is 1, chromosome 1, chromosome 2, chromosome 3, chromosome 4. So 1 with 3, then 1 with 4. Then 2 with 3, 2 with 4. You'll get the 4 gametes. 4 possible gametes from a heterozygous individual that's heterozygous for the 2 characteristics. Okay, so to summarize what we discussed in the previous slides, dihybrid inheritance involves a cross between 2 parents that differ in 2 characteristics, such as height of plant, either tall or dwarf. So this is the first characteristic, height, and the second characteristic may be shape of seed. All right, and the traits may be round or wrinkled seed. Wrinkled, in the textbook they use the word constricted instead, okay, but it means wrinkled. So here are examples of 2 crosses. We are going to discuss the first one. A purebred tall plant with purple flowers is crossed with a purebred dwarf plant with white flowers. So just now we've discussed already in the previous slide. The characteristics are height and flower color. Okay, and you have the 4 alleles here. Tall trait determined by allele big T, which is dominant, and dwarf trait determined by allele small t, which is recessive. Purple flower trait determined by allele big R, which is dominant, and white flower trait determined by allele small r, which is recessive. Now the textbook has this example which is discussed in the textbook. So a purebred plant that has round yellow seed is crossed with a purebred plant with wrinkled green seed. Okay, so if let's say you read this type of this description in a question, you have to identify whether it's monohybrid or dihybrid inheritance, right? So then you have to see here the round and wrinkled seed traits. They concern the seed shape. So that's one characteristic. Then yellow and green seed. So this is about the characteristic of seed color. Okay. So as an example, we can assign letters like big B. So round seed trait determined by allele big B, which is dominant, and wrinkled seed trait determined by allele small b, which is recessive. For seed color, yellow seed trait determined by allele big K, which is dominant, and green seed trait is determined by allele small k, which is recessive. Now here's an example of a dihybrid cross, okay, for learning standard 2, and it's similar to the cross that Mendel did in his experiments. So pure breeding tall pea plants which produce purple flowers were crossed with pure breeding dwarf pea plants with white flowers. So here the two characteristics are height and color of the flower. So the pure breeding plants were crossed between each other, and they have contrasting traits. Now, results. All of the F1 generation offspring were tall plants with purple flowers. Okay, so from here we know that the tall trait and the purple flower trait are the dominant traits compared to the dwarf trait and the white flower traits. Now, after that, the F1 hybrid plants were self-crossed. They're heterozygous hybrid plants. They were self-crossed. That means self-pollinated, pollinated among themselves, and then self-fertilized. Then after the seeds were obtained, these were the F2 plants. They were planted, and these are the F2 plants. So after they mature, we can find that the F2 plants had 4 different phenotypes. Okay, like for example, tall plant with purple flower, then tall plant with white flower, and so forth. And the phenotypic ratio is 9:3:3:1. Think of 9 equals to 3 times 3 times 1. So this ratio is different from the monohybrid ratio of 3:1 when 2 heterozygous plants are crossed. This is 9:3:3:1. So you're going to find out how we got this ratio, okay, from the cross, from the dihybrid cross. Now let's look at the schematic diagram for the experiment that we are discussing. So parental generation are purebred tall plants with purple flowers, and this is crossed with the purebred dwarf plant with white flowers. All right. So both parents have contrasting traits, and they are purebred. Now the F1 phenotype, F1 that is produced have this phenotype. All are tall plants with purple flowers. All right. And our conclusion is that the tall stem and the purple flower traits are dominant, okay, compared to the dwarf trait and the white flower trait, right? A phenotype means the observable characteristic. Now, after that, there was a self-pollination of the F1 plants to get the F2 plants, and the F2 phenotypic ratio is 9 plants which are tall with purple flowers is to 3 plants which are tall with white flowers is to 3 plants which are dwarf with purple flowers is to 1 plant which is a dwarf with white flowers. And Mendel was able to explain how he got this phenotypic ratio of 9:3:1. So we're going to discuss these two. Okay, so here's the important part. According to learning standard number 3, we must be able to construct a dihybrid cross diagram like the one shown here, and also be able to explain it. All right. So the dihybrid cross diagram is different from the monohybrid cross diagram. Now, explanation of the experiment on dihybrid inheritance. First, write down your key. Okay, let big T represent the dominant allele for tall trait and small t represent the recessive allele for dwarf trait. Secondly, for flower color, let big R represent the dominant allele for purple flower trait and small r represent the recessive allele for white flower trait. Okay, now next, parental phenotype. So here we have a list of the titles that you have to write on the left hand side. You must memorize all these titles, must write it in, and then write the corresponding parts here. Okay, like for example, parental phenotype. Okay, on the same line you can write purebred tall purple flower plant and purebred dwarf white flower plant. Okay, the phenotype is the observable characteristic. Okay, but what is the genotype? Meaning the genetic content inside, genetic composition. Okay, and that is what are the alleles in each diploid cell of the plant. Okay, so for the tall plant with purple flowers, since it's purebred, it must be homozygous dominant for both characteristics. That means it has 2 big T alleles and 2 big R alleles. Okay, so you must have allele big T to determine that it's tall. You must have allele big R to determine that it has purple flowers. Okay, but it's homozygous for both the traits. Okay, so big T, big R, big R. Okay, having 2 identical big T alleles and 2 identical big R alleles. Similarly, for the dwarf plant with white flowers, and in contrast, it has the recessive alleles only. Okay, so it's homozygous recessive for the trait of dwarf stem, and also homozygous recessive for the trait of white flower. It has 2 identical recessive alleles, okay, small t, and 2 identical recessive alleles, small r. All right. Now next, in meiosis, through meiosis, gametes are formed. Okay, so how do we determine the alleles in the gametes? Follow Mendel's second law. Okay, referring back to the diagram of the cells that we saw just now. Now we use the link method, and that is, according to Mendel's second law, any allele from the first pair of alleles here, big T, can combine with any allele from the second pair of alleles, which is big R. Okay, so big T can combine with big R. We link it together like that. So you get big T, big R, or the first big T with the second big R. You still get the same result. Then use the second big T with big R, big T with big R. So all of the gametes have the same combination of alleles, big T, big R. Okay, so you must take up 1 T and 1 R. So this is called the link method. To determine, my way of saying is the link method, to determine the alleles in the gametes. Okay, and we can also look at it this way. If there are 4 alleles here, numbered 1, 2, 3, 4, so you can combine first allele with third allele, 1 with 3 or 1 with 4, and then 2 with 3 or 2 with 4. Okay, in this case, all the gametes have the same alleles, so we just draw 1 is enough, 1 representative of it. Then for the dwarf plant with white flowers, again we use the link method and Mendel's second law to determine the alleles in the gametes. So small t can combine with small r, small t with small r, and this second small t with small r, small t with small r. So all the gametes have the alleles small t, small r. So just write 1 enough. Then in fertilization, the gametes will fuse together and form the zygote, right? Okay, which will be the offspring. So every cell in this offspring will have this combination of alleles. Okay, so combine them together, all the alleles here, but always write the T's together and the R's together. So big T, small t, big R, small r. So this is the genotype of the F1 generation, which is the offspring of the parents, right? Which are the offspring of the parents. And here, this plant is tall with purple flowers, since it has the dominant allele for tall stem trait, big T, and has the dominant allele for purple flower trait, which is big R. Okay. However, it is not the same as this parent here, because it is heterozygous. It does have the recessive alleles too. Okay, unlike this parent here. Okay, this first parent. So only dominant traits are expressed in the heterozygous F1 plant. Why? Because the dominant allele will suppress the effect of the recessive allele, and then the dominant allele will express its trait. Okay, so all the dominant traits will be expressed. in this plant. That is, the plant will be tall because of the dominant allele for tall stem trait, big T, and the plant will have purple flowers because of the dominant allele, big R, for purple flower trait. Okay? Right, so this is the first part of the experiment. Now we're going to go on to the second part of the experiment. Now here are the notes for the explanation of the cross between the two parents. All right. So Mendel carried out a dihybrid cross to explain how two different characteristics were inherited. For example, a cross between two purebred plants can be done to study the two characteristics of pea plants. So the two characteristics studied are height of plant, either tall or dwarf, and the color of the flower, either purple or white. So to begin, each purebred parent has two different pairs of alleles, and the alleles in each pair are identical. Now, since parent A is purebred, it is homozygous dominant for traits of tall stem and purple flower. So this is its genotype, big T, big T, big R, big R. It produces gametes with alleles T, big R through meiosis. Okay, as you have seen just now, the gamete has alleles big T, big R. And the second parent is parent B. It is homozygous recessive for the traits of dwarf stem and white flower. So it has two identical recessive alleles for dwarf stem, big small t, and two identical recessive alleles for white flower, small r. Okay, so it produces gametes with alleles small t, small r through meiosis, as you've seen just now. Okay, then fertilization between the gametes with alleles big T, big R, and alleles small t, small r from the two purebred parents will produce the F1 generation, which is a hybrid, which are hybrids, and they have the genotype big T, small t, big R, small r. Okay, which all have the dominant traits of tall stem and purple flower. Then recessive traits of dwarf stem and white flower are not observed in F1 generation, and that's because the tall stem trait is dominant compared to the dwarf stem trait. The purple flower trait is dominant compared to the white flower trait. Okay, now let's go on to the second part of the experiment where the hybrids were crossed among themselves. Now big T, big R, small r, they were crossed among themselves. Now we've just discussed the cross between two parents to produce the F1 generation. So now let's discuss the self-cross of the F1 plants to produce the F2 generation plants. Now the F1 phenotype is tall plant with purple flowers, and similarly for the other parent too. Then its genotype. So this plant is actually a dihybrid, meaning it's heterozygous for both characteristics, for the characteristic of height and flower color. Okay, so the genotype is big T, big R, small r, and it's the same for the other parent, right? Then through meiosis, gametes are formed. So what are the alleles in the gametes? Now, according to Mendel's second law, during formation of gametes, any allele for height can pair up with any allele for flower color. Any allele for the first characteristic can pair up with any allele for the second characteristic. Okay, so big T can combine with big R. So you get the gametes, a gamete that contains the alleles big T, big R, or big T with small r. Okay, so the second gamete has alleles big T, small r. Then small t can combine with big R, and then small t with small r. Okay, so altogether, four different gametes are formed, four different types of gametes are formed from one parent, and it's the same for the other parent. Right, how to determine the genotypes of the F2 generation? So the first gamete from the first plant can fuse with the first gamete from the second plant, right? So normally we'll draw the line down here and the line down here. Then we will combine all the alleles together, all right? But if you do this method, you will have to have 16 pairs of arrows because you have 4 gametes from the first parent, 4 gametes from the second parent. So there will be 16 combinations for the genotypes. So it's not possible to do it. It's too complex. If we use the crisscross method. So we use the Punnett Square for the hybrid inheritance, in which you draw a 5 by 5 table, 5 columns and 5 rows. And then the first column is a bit broader because you have to write down the male gametes, the title male gametes. So all the male gametes are written here. Now transfer from the gametes here. Just write down the alleles for each gamete. So big T, big R. Then this one, transfer here, big T, small r. Small t, big R, and small t, small r. Okay. Similarly for the other parent, which produces the female gametes. So write down all the alleles in the gametes here. Alright. Then determine the genotypes of the F2 generation. So you can draw your table first. Practice drawing this table and determining the genotypes in the boxes. Okay, here. Then pause the video while you're doing it, and then after that, check with the answers here. What are the genotypes of the F2 generation? So I hope you have already written down the genotypes of the F2 plants in this space here. For example, if you want to know what's the genotype of this offspring here, then you just look up. You'll see that the male gamete has alleles big T, small r, and look horizontally this way. Okay, you'll see also big T, small r, the alleles in the female gamete. All right. So combine them together, and there you have the offspring, right? So this is the genotype of the offspring from the fusion of this male gamete with this female gamete. So put all the T's together, big T, and then all the R's together. Small r, small r. Okay, don't write as big T, small r, big T, small r. Okay, that's not correct. All right. Okay, so after you have determined the genotypes of the F2 generation, you have to determine the phenotypes. Okay, from the genotypes here we determine the phenotypes. Okay, so try and determine the phenotypes of the F2 generation. We are going to group them together. So there are 4 different phenotypes. Let's have a look at the next diagram. Now the next step is to identify the phenotypes of the F2 generation plants. Okay, so you have already obtained the genotypes of the F2 generation. Now let us identify the phenotypes. All right. So this cross is between 2 heterozygous plants, the F1 generation plants, and these are the F2 genotypes. Now find the plants that have this phenotype: tall with purple flowers. So actually there are only 4 phenotypes that you can obtain. All right. Tall purple flower, tall white flower, dwarf purple flower, and dwarf white flower. So now we want to find out whether the ratio here of these 4 types of phenotypes meet the 9331 ratio. Okay, it's the same as the 9331 ratio or not. All right. So let's try to find all the plants which are tall with purple flowers. So you can pause the video for a while, try and find the plants with tall purple flowers, then tall white flowers, dwarf purple flowers, and dwarf white flowers, and then mark it in your diagram. For tall purple flower, we use the symbol, the asterisk. For tall white flower, we use the hash symbol. For dwarf purple flower, I use the inverted V symbol. And for dwarf white flower, I use the X symbol. Okay, so you can try to do it yourself first by marking it in the boxes here, and then we will discuss it in a short while. So pause the video. Okay, let's look at the answers. So tall purple flower plant should have at least 1 big T and 1 big R in order to have this phenotype. Okay, so you can see here that if the male gamete has big T, big R, then all the offsprings that are formed from the fusion of this male gamete with the female gametes will have at least 1 big T, 1 big R. So all these are tall with purple flowers. All right. Likewise, this way too. There's a female gamete with the alleles big T, big R. So all these offsprings here are tall with purple flowers, right? Having at least 1 big T and 1 big R. And also in this diagonal form, in this the offsprings that are formed here. Okay, they form a diagonal line. They are all heterozygous. So there are 4 heterozygous plants that are heterozygous for the 2 characteristics or the 2 traits, right? So this one you can see is big T, small t, big R, small r. And likewise for this and this and this. So at least one big T and one big R. So they are also tall with purple flowers. So they're all together 9 plants. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 plants which are tall with purple flowers. Okay, from the Punnett Square here. How about the tall plants with white flowers? So to have white flowers, they must be homozygous recessive for the trait of white flower. Because trait of white flower is a recessive trait controlled by a recessive allele. So the plant must be homozygous recessive in order to express this trait of white flowers. Okay, and at least one big T. Okay, for the trait of tall stem. So you can mark the plants with the symbol H. Okay, and there are 3 of them here. Now at least one big T and 2 small r. Likewise here and 1 here. A bit scattered. Next are the plants which are dwarf with purple flowers. So if you search from this Punnett Square, you'll see there are 3 of them. Okay, for the trait of dwarf stem, the plant must be homozygous recessive for that trait. Okay, so having 2 recessive alleles, small t, okay, and at least one big R, so that it will be able to express the purple flower trait. Okay, so there are 3. And lastly, the plant with 2 recessive traits, dwarf and white flowers. Okay, there's only 1. So all the alleles here are recessive. Small t, small r, small r. Okay, so if you count them, you will find that the numbers for each plant, each phenotype, you'll find that the ratio is 9, 3, 3, 1. Okay, remember 9 equals to 3 times 3 times 1. Just to help you remember 9, 3, 3, 1. So this is called the dihybrid ratio. It is obtained when 2 plants which are heterozygous for both characteristics are crossed. It's called the dihybrid ratio. So here's the explanation for the cross between the 2 F1 plants, explanation of dihybrid inheritance. We just continue with this, the cross between 2 F1 plants, or between the F1 plants, big T, small t, big R, small r. I'll just read from here. The heterozygous F1 generation plants produced from the parental cross were self-pollinated and self-crossed. Using Mendel's second law through meiosis, there are 4 possible combinations of alleles in the male and female gametes of the heterozygous F1 plants. In the gametes, so the alleles may be big T, big T, small t, big R, and small t, small r. Now, because fertilization is random, any male gamete can fuse with any female gamete. And there are 16 possible combinations of gametes in the production of the F2 genotypes, as shown in the Punnett Square, which is a 5 by 5 table. But there are actually only 9 different genotypes in the F2 plants. We'll see that in the next slide. 4 different phenotypes were produced in the dihybrid ratio of 9, 3, 3, 1. The ratio is as follows. 9 with 2 dominant traits, that means tall and purple, with purple flowers. 3 with 1 dominant trait and 1 recessive trait, that is tall with white flowers, right? And then 3 with another dominant trait and another recessive trait. That is dwarf with purple flowers, right? The purple flower trait is the dominant trait and the dwarf trait is the recessive trait. And 1 with 2 recessive traits, that means dwarf with white flowers. Okay, so 9, 3, 3, 1 ratio. So we can group the F2 genotypes together according to the phenotypes, all right, from the cross between 2 dihybrids or heterozygous plants. All in all, there are actually only 9 genotypes as shown here. They're all different. These genotypes are all different. And their phenotypes, there are only 4. Okay, there are a total of 4 phenotypes. For example, tall purple flower plant will have either this genotype or this one or this one or this one, and so forth. So this is the analysis of the Punnett Square table. All right, this is just for your information and may help you in understanding the topic better. Here's something to take note. In dihybrid inheritance, 9, 3, 3, 1 is the dihybrid ratio produced when 2 heterozygous dihybrid plants are crossed. That is big T, big B, small b, crossed with big T, big B, small b, as an example. Okay, so 9:3:3:1, this ratio is called the dihybrid ratio for the hybrid inheritance. But 3:1 is the monohybrid ratio produced when two heterozygous monohybrid plants are crossed. That is big T crossed with big T small t. So these two ratios must be remembered, especially for the problems, questions involving genetic schematic diagrams. Finally, as a conclusion, let's look at Mendel's second law. So Mendel introduced Mendel's second law, or law of independent assortment, okay, which is related to dihybrid inheritance. It states, during gamete formation, each allele from a pair of alleles, such as big T, small t, can combine randomly with any allele from another pair of alleles, such as big B, small b. This is for dihybrid inheritance. For example, if you have a plant with this genotype, okay, it can be any genotype for dihybrid inheritance, then you can determine the alleles in each gamete using Mendel's second law, which we have already applied just now to determine the gametes, right? Okay, for the experiment that we were discussing. So, for example, any allele from this first pair of alleles, big T, can combine randomly with any allele from the second pair of alleles, which is big R, small r. So big T combines with big R, you will get this gamete with this combination of alleles, and then big T with small r. So the second gamete has the alleles big T, small r. Now the second one, second big T with big R, and second big T with small r. So actually there are only two types of gametes formed here if you look at the contents, the genetic contents inside the gametes, and that is the gamete with big T, big R. These two are the same, and the gamete with big T, small r. Okay, so what's the biological reason for Mendel's second law? This is because Mendel's second law holds true because we know that now, during last time Mendel didn't know about it, but now, okay, we can explain his law well. During meiosis, different genes, meaning different pairs of alleles, found on different separate chromosomes, separate homologous chromosomes, will assort independently of each other, just as the chromosomes assort independently too, all right, when they are arranged on the metaphase plate and they separate during anaphase 1 of meiosis. Assort means to separate. Due to the random arrangement of chromosomes on the metaphase plate in metaphase 1 of meiosis, the random arrangement of homologous chromosomes. They arrange themselves in different ways. That means the chromosomes, homologous chromosomes, are arranged independently of each other. All right. So there are a few possibilities that can happen in the combination of the different homologous chromosomes. Now, application. Mendel's second law is used to determine possible combinations of alleles in gametes for dihybrid crosses, as we have done here. All right. So try this dihybrid. Try this question. What are the gametes for the plant with the genotype big B, small b, big N, big N? All right. So apply Mendel's second law and then determine the gametes, and pause the video for a while. Then you can look at the answer after that. Okay, let's write the answer. So the gametes that you should get will be big B, big N, with the alleles big B, big N, and the gamete with alleles small b, big N. Okay, there are only two types of gametes formed. All right, using Mendel's second law here, big B can combine with big N, and then big B with big N again, and then small b with big N, small b with small n again. So these are the two different types of gametes formed, okay, with different combinations of alleles. Now it's important to know the assumption to make for Mendel's second law to be true. All right. So for Mendel's second law to be true, the two different pairs of alleles that control the different characteristics must be on different types of chromosomes and not on the same type of chromosome. For example, like this, as we have seen before, this first pair of alleles, the B alleles, are on a pair of homologous chromosomes, number 1. where there's one paternal chromosome and one maternal chromosome, right? But the second pair of alleles is on another pair of chromosomes, homologous chromosomes number 2, different from homologous chromosomes number 1. Then Mendel's second law of independent assortment will hold true, okay, where the two pairs of alleles will separate independently of each other. Two pairs of chromosomes will separate independently of each other, and so will the two pairs of alleles. So it's not like this situation. If the situation is like that, where the first pair of alleles, the B alleles, are on chromosome number 1, and the second pair is also on chromosome number 1, maybe at a different loci. Then during the formation of gametes, there's a greater likelihood that the big B and big A alleles will be found together, and the small b and small a alleles are found together, unless of course there's crossing over and then exchange of segments of DNA and so forth. Then you may also get the combination where big B is together with small a in the gamete that's formed, or small b with big A. Okay, but the probability will not be equal for all the gametes, the 4 different types of gametes that are formed, as we have seen for this case, where the 2 pairs of alleles are on different types of chromosomes. Okay, so for Mendel's law to be true, this is the assumption we have to make. Okay, and not this one. Let's look at 3 questions on dihybrid inheritance. You can pause the video for a while and try the questions yourself. Then we'll discuss it. Right, now let's look at the questions. What type of gametes can be produced by a garden pea plant that is heterozygous for flower position and seed shape? Big A, small a, big R, small r. This is the genotype of the pea plant that is heterozygous, right? So take note here, now underline these words, type of gametes. Okay, they want to know about the gametes, not the offspring of the plant. So what type of gametes can be produced by the pea plant with this genotype? Okay, so straight away we just apply Mendel's second law and also use the link method to find the alleles in the gametes. So Mendel's second law says that any allele from the first pair of alleles can combine with any alleles from the second pair of alleles. Okay, so big A combined with big R, that will be in the first gamete. Big A with small r, so these 2 alleles will be in the second gamete. Small a with big R, small a with small r. So the answer is C, right? Now D is not correct because you have one gamete with 2 R alleles, big R and small r. There's no allele, no A allele, so it's wrong. Because you have one copy of each type of allele. A allele 1 copy and R allele 1 copy in a gamete. Number 2, a garden pea plant is heterozygous for both height and seed color. What percentage of its gametes would be expected to contain both the dominant allele for tall plant and the dominant allele for yellow seed? Okay, so again highlight the keywords here, heterozygous, height and seed color. Percentage of gametes containing dominant allele for tall stem and dominant allele for yellow seed. Okay, so the first thing to do is to assign letters to represent the alleles. For height, let big T represent the allele for tall stem, which is dominant, tall plant. And small t represents the allele for dwarf plant or dwarf stem, and it is recessive. For seed color, we can use big R to represent the dominant allele for yellow seed, and small r to represent the recessive allele for green seed. Okay, so now this plant is heterozygous, so it should have this genotype: big T, big R, small r. All right. Then the question wants to know what percentage of its gametes will contain one dominant allele for tall plant, which is 1 big T, and one dominant allele for yellow seed, which is 1 big R. Okay, so do the use Mendel's second law to determine the gametes again, and use the link method. Big T can combine with big R. Okay, or big T can combine with small r. Okay, so these two alleles will be in the second gamete. Small t with big R, small t combines with small r. Okay, so we have 4 different types of gametes, but only one which has the dominant allele for tall plant, big T, and dominant allele for yellow seed, which is big R. Okay, so 1 out of 4 gametes. So how many percent is that? A quarter times 100 equals to 85%. All right. Now, next question. The diagram above shows a cross between 2 plants, plant 1 and plant 2, in a study on dihybrid inheritance. State the genotypes which are numbered 1, 2, and 3. 1, 2, and 3. So for this question, first of all, we determine the gamete, the alleles in the gamete. Big B, small g, small g. Okay, so using Mendel's second law again, big B can combine with small g, big B with small g. Then the other one is also big B with small g, big B with small g. So all of them have the same, the gametes will all have the same alleles inside. And for number 2, the answer is big B, small g. Okay, next, the F1 generation is formed when the gametes from plant 1 fuse with the gametes from plant 2. Okay, so it's a heterozygous organism here. And let's remove the gametes, the alleles, remove the alleles that is used to form this gamete here. Okay, so remove big B and remove small g. That will leave you small b, big G here. Okay, and then for plant 2, it should have 2 copies of the alleles of each type of allele. So small b with big G, big G. Okay, that will be the answer. Okay, let's write down the answer. Genotype 1, small b, big G, big G. Genotype 2 is for the gamete here. Genotype 3 for the other gamete here. Okay, so that's all for this lesson. Thanks for watching. Please do share, like, and subscribe. So have a good time revising for your SPM, and take care of your health. So all the best in your studies till we meet again.